Also.
Chemistry labs. Standard chemical laboratories exist for a variety of purposes. Dissolution and purification of different raw materials by means of standard chemical procedures. Development and modification of new polymeric structures, membranes, composites, hydrogels, by means of chemical and enzymatic approaches. Production of microspheres and fibers by wet spinning technique. Production of nanofibers by means of electrospinning. Study of behavior of the natural polymers in simulated physiological solutions and degradation mechanisms.
We are in the General Chemistry Lab, where we can prepare several different materials, and also we can process these materials by different equipments which we have. And we have the plasma reactor, which allows us surface modification of different materials.
The Bioreactors Laboratory is a facility specifically devoted to the development and functionality assessment of advanced culturing systems. That is, systems that have the potential to enhance the functionality of tissue-engineered constructs as compared to the standard static culturing methodologies.
We are in the Bioreactors Lab, where we develop advanced culturing system in order to obtain tissue-engineered constructs with high functionality. We try to develop systems that can enhance cell proliferation and distribution within the construct, but also to provide mechanical stimulus like compression or fluid shear stresses that can enhance stem cells differentiation in these systems.
All the experiments involving cell lines, primary cells and/or stem cells after isolation from different sources are performed in the cell culture laboratory, from basic research to tissue engineering studies using the different materials developed in the group. The Cell Culture Lab is equipped with all the standard cell culture equipment, including 6 biohazard laminar flow chambers, 4 CO2 incubators, an inverted light microscope and 2 centrifuges. The Cell Culture Lab is a cleanroom facility that has specific operating rules, and therefore only certified researchers are allowed to work there.
Basically, we combine materials, and we always use materials of natural origin with stem cells. Stem cells that we always use cells obtained from the patient that we want to treat. So in this case, we are seeing some cells from umbilical cord that we have been isolating from umbilical cords that we receive from local hospitals. And then we culture those cells in contact with different materials, and if everything goes okay, the cells are going to be differentiated to the tissue that we want to engineer in the lab, and the degradable scaffold that we use to culture the cells is going to be degraded during time, and by the end of the day what we have is a tissue of the same patient, and the biodegradable polymers was already degraded and is not in there anymore, so everything works as we would like.
The Microscopy and Flow Cytometry Laboratory allows the preliminary structural analysis of biomaterial specimens, such as 3D structures for tissue engineering applications, obtained from different processing methodologies, and the follow-up of its subsequent biological in vitro and in vivo characterization. The reproducible microscopic examination of the biological performance of the developed biomaterials, as well as of the diverse primary and stem cell populations isolated from several sources, can be accomplished with bright field, using either transmitted or reflected light, and fluorescence after appropriate cell staining. A full and complementary characterization of cell subpopulations of interest can be carried out by multiparametric flow cytometry analysis.
This is our microscopy laboratory, where we have a flow cytometer that we can use to characterize the stem cell populations that we isolate from the different sources, and also our fluorescent microscope where we can see and analyze all the data coming not only from the in vitro tests, but also from the animal studies.
The Histology Laboratory is equipped with a cryostat, a tissue processor, a paraffin embedding center, a microtome, and automatic staining equipment.
This is our histology lab that has all the equipment necessary to histologically process the samples that come not only from the cell culture laboratory but also from the animal studies.
The biologic characterization and PCR laboratories are equipped with a microplate reader, 2 cryopreservation tanks, horizontal and vertical electrophoresis tanks, transilluminator, UVP, together with other small equipment. This equipment allows the characterization of the cultured cells. Additionally, this laboratory supports the cell culture laboratory for reagent preparation. The PCR laboratory is equipped with a flow chamber, a nanodrop, and a thermocycler. With the available equipment, it is possible to perform the extraction and purification of nucleic acids, the quantification of protein and nucleic acids, and also the expression and quantification of particular genes on biological samples.
This is our biology laboratory, where we do the biochemical and molecular analysis of the constructs that we obtained in the cell culture laboratory, and that serves basically as a support laboratory for all the biology wing of our building.
The animal facilities are located in the underground of the building and have a total area of around 200 square meters. These facilities allow the housing of immunocompetent and immunocompromised rodents, mice and rats, and lagomorphs, rabbits. Additionally, it is possible to breed rodents. The equipment available allows setting up surgical procedures aimed at testing the performance of the assembled constructs for different tissue engineering applications. Through our cooperation with the Veterinary School of Trás-os-Montes e Alto Douro University, we can set up experiments with medium-sized animal models such as goats.
What you are seeing here in the image is that we performed an experiment. In fact, this was in collaboration with a partner in Israel, Libn, and we implanted in nude mice either our materials with human bone marrow stem cells differentiated into the osteogenic phenotype, or left the defects empty or implanted with our porous materials. And I'm sure you can appreciate the difference that we have after 6 weeks of implantation when we use the defect without any material, with our material, and with human bone marrow stem cells. And you can really see that the recovery of the defect after 6 weeks, it's much better when we use our materials in combination with human bone marrow stem cells.
The group has been able to attract students from approximately 20 different countries. All the PhD students have been working partially in these laboratories, within the framework of the collaborations established. Such connections have been reinforced through the international consortia formed in the context of European projects, most of them coordinated by the group. Solid collaboration with international groups has been a main strategic objective since the outset. The 3B's research group was selected to organize the European Congress of the Tissue Engineering and Regenerative International Society, TERMIS, in 2008. The third annual conference of the European section of the TERMIS, TERMIS EU 2008, was held in the Porto Congress Centre, Alfândega, between the 22nd and the 26th June 2008, with approximately 700 participants and experts from around the world, and discussing 750 scientific papers in the field of tissue engineering. This achievement exceeded all expectations. In the scope of the Marie Curie actions, and as mentioned earlier, the 3B's research group also coordinated INVENTS, an integrated series of events for high-level training on biomaterials, tissue engineering, controlled drug delivery and related emerging fields, which ended in December 2009. This event sought to organize a series of events with high-level scientific training, including practical courses in areas such as tissue engineering, controlled release of drugs, biomineralization, biocompatibility, self-assembly at a nanoscale, and stem cells. This group, through Professor Royel Reich, has been responsible for the organization of several other principal international meetings, workshops and conferences held in many different locations in Portugal and other countries. Royel Reich is the editor-in-chief of the Journal of Tissue Engineering and Regenerative Medicine, Wiley-Blackwell. He has also edited several books for the international market, as well as several scientific journal special issues. This is one of the groups in the world with the strongest publication outputs in the different areas related to biomaterials, tissue engineering, and regenerative medicine.
We have our own journal that we are in charge of it. I am the editor-in-chief of this new journal. It is called the Journal of Tissue Engineering and Regenerative Medicine, and is published by one of the main publishers in the world, which is Wiley-Blackwell, and it's going very well.